My name is Barbara. I'm from the Institute of Systematic and Evolutionary Botany. And today I will tell you not to how to generate a high-quality reference genome, but how it can be used to aid assessing genetic diversity and conservation of an endangered species in Switzerland. As you all know, we are in the midst of a biodiversity crisis, and freshwater biodiversity is especially hard hit, as you can see on the figure on your right top corner. Maybe because aquatic habitats have degraded rapidly, many have been lost, water has been polluted, and the flow regime has been changed. The study species I'm working with is Hottonia palustris. It's a heterostylous freshwater species. In such species, populations are composed of two different types of plants. One type of plant produce long-styled flowers with short anthers, and the other type of plant produce short-styled flowers and high anthers. This is a floral polymorphism that is optimized for outcrossing, as pollen transfer between the two self-incompatible morphs is optimized. As most aquatic plants, Hottonia palustris can also reproduce clonally. And important for the species is now that if only one morph is left in a population, then sexual reproduction is highly unlikely and clonal reproduction may prevail. Hottonia palustris occurs from West Asia to Europe in shallow, mesotrophic, semi-shaded ponds and lakes. In Switzerland, the species has sharply declined from 1930 till today, mostly due to the loss of habitats and isolation of suitable habitats. As you know, eutrophication and water regime changes have lost many aquatic habitats. In many populations, we have few and very unfit plants left, as you can see on the picture. They're the very poor Hottonias floating around. In addition to that, Hottonia palustris produce super nice flowers. Everybody wants to have them in their garden pond, so you can buy them actually in garden centers. The question is now, how do these garden center plants affect the natural biodiversity in Switzerland and other countries? So do we have populations that are composed entirely of garden center plants? Do we have mixed populations with garden center plants and natural plants? What is the genetic diversity and the fitness of those populations? One important note about these naturalized garden center plants is the effect on biodiversity they have depends strongly if they were collected in spatially close or distant source populations. How long were they cultivated ex situ for few or many generations, and were they propagated clonally or sexually? Therefore, in this applied study, we address the following questions. Were populations stocked with plants from garden centers, or the other way around, from which natural populations were garden center plants taken? And what is the genetic structure and fitness of the natural populations? To do that, we collected plants from 26 Swiss populations, always 3 plants per population if possible. And as outgrouped, we collected a population in France and 1 in Germany. In addition to that, we included plants from 4 local garden centers. I don't have to tell you that, but reference-based approaches are the best to use. So, as reference genomes act as a filter, it's very important to use high reference genomes, and ideally from the target species, especially if one looks at fine-scale variation as we did in our study. And here you see our high-quality reference genome that we did with ERGA. Now, we were working with aquatic plants, and there the reference genome is even more important as the leaves are submerged, so they're actually covered by all varieties of organisms, and therefore the reference genome is crucial to actually exclude these contaminants in the sequencing data. We used the ddRAD sequencing and the reference-based stack pipeline to generate 27,000 SNPs in 94 individuals. To the results. We analyzed the data in different ways. Here in the middle, you see the phylogenetic tree, and on the right side, you see the structure results. The first question was, did we find stocking of non-native plants or garden center plants? The first important result is that we found two main clusters. One cluster that included the French, German, and most garden center plants and very few Swiss plants. And the second cluster that contained the most Swiss plants and one garden center plant. One word to this garden center plant, we contacted the owner of the shop and he confirmed that he recently collected plants from a natural Swiss population, and therefore it makes very much sense that this garden center plant actually was in the same clade as the source population. The other plants that cluster in the French and German clade, as you can see here on top, these are the plants or the populations, and then below you see the map where they occur. We concluded that these 6 populations were stocked because we had such a high differentiation between the two clades. So these are populations which were stocked with garden center plants that did not originate from Switzerland. They were maybe collected somewhere north of Switzerland in Germany. In addition to that, we found very little variation in the clades, in all the clades from the plants that we collected in Switzerland, and very high variation among the Swiss clades. Compared to the French on top and the German plants, they were much more differentiated. This is also visible when we look at the structure result. The French and the German plants, each dot represent one plant, are more diverse than the Swiss plants, where we can see below. In addition to that, we didn't find a clear geographic structure. The plants that were assigned to the cluster 1, the blue cluster in structure, they occur all over Switzerland. Second step, we looked at the genetic structure of the populations, and we found that some populations here indicated in red are very high differentiated, while others were very similar. Indeed, we found 5 sets of populations that were almost identical, with almost zero genetic differentiation. After that, we zoomed in at the individual level and looked at the genetic distance between individuals. Again, we saw that some individuals were highly distant to each other genetically, others in blue were very similar. In fact, we actually identified 12 clonal lineages in our data set. And when we look where these clonal lineages occur on the phylogenetic tree, I hope you can see the colors. Then in each clade, 1 or sometimes even 2 clonal lineages were present. The fitness of the populations we looked at by comparing the heterozygosity among the populations or the genetic diversity, and it was quite variable among the populations. Some had very low heterozygosity and some actually here you can see very high, but was very important that we find an excess of heterozygosity in 19 of the populations, which indicate again that there is clonal reproduction. Similarly, when we looked at the inbreeding index in the same population, this inbreeding index was highly negative, which is again an indication of clonal reproduction. Back to the biology of the species. As I said in the beginning of my talk, both morphs, floral morphs, must be present in a population so that they can reproduce sexually. And if only one morph is present, then sexual reproduction is very unlikely. Here you see the result of the clonal composition of the populations. We found only both morphs present in France and Germany, while all Swiss populations either had only long-styled morphs in red or short-styled morphs in blue. Therefore, sexual reproduction is unlikely to occur in Swiss populations. To summarize, we found that 6 populations were most likely stocked with non-native plants. All lines of evidence suggest that sexual reproduction is scarce in Switzerland, so clonal reproduction prevails. And since the clades are very highly differentiated, hence preserved unique genetic diversity, the clades should be conserved. To foster sexual reproduction, one could try to stock populations with long and short style plants and see what's the outcome. Do they produce fit offspring, or there might be the chance or that there is actually some outbreeding depression. To conclude, we can say that Hottonia palustris is an endangered heterostylous freshwater species with an unsecure future, at least in Switzerland. With that, I have to thank Charlotte, Agnes, Wolfgang, Annin, and Rolf for help in the fieldwork, and the different cantons and the University of Zurich for funding, and thank you for your attendance.
So now again, open for questions. If anyone, you can put it in the chat or open your microphone. There is one here from Christian asking, before having a high quality reference genome, did you try the de novo approach and stacks? If yes, did you compare the differences in capturing fine scale genetic structure between the two methods?
Yes, we did that. So we did both in parallel because we had the reference genome before at hand. But with the de novo approach, we had much more missing data. We already had a lot of missing data, and the pattern was less clear. So we had a less good separation and less clear patterns. So the reference genome helped a lot to identify reliable SNPs in the data set.